A detailed look at the flu vaccine, covering how it's created, some common problems, and its effectiveness. The process begins by taking millions of fertilized chicken eggs and inserting each with a candidate vaccine virus. This process can create either the standard injected killed vaccine or the live weakened vaccine in a nasal spray. The inserted candidate vaccine's viruses are left to incubate for several days to allow for replication. This process will take place all over the world. In the US, we use privately made influenza vaccines. The candidate virus is then removed from the egg. For the vaccine shot, the virus is killed and a purification process begins to isolate the virus. For the nasal spray, the virus is weakened and goes through a different process. Once the virus has been purified and approved by the FDA, it is placed into a vial and sent out to the distributors. There are a total of 3 approved methods for creating a flu vaccine: the aforementioned chicken egg method, a cell-grown method, and what is called the recombinant method. The cell-based method uses the recommended candidate viruses by the World Health Organization. The candidate viruses are then inoculated into a cultured mammalian cell. They are left for several days to incubate similarly to the egg-based method. The virus is then removed and follows a similar path to the egg-based method with purification and FDA testing. This method is quicker than the egg-based method and doesn't require millions of eggs. The recombinant method starts with a naturally occurring vaccine virus. The HA protein that induces an immune response in humans is isolated from this virus. This isolated protein is then combined with another virus that grows well in insects. This mixture is then mixed with insect cells and given several days to replicate. The HA protein is again harvested from the cells and purified. Once approved, it is packaged and sent to distributors. This is the quickest method for creating a flu vaccine. Problems. It is commonly cited by people and the CDC that a big reason for the lack of effectiveness with the vaccine is the fact that the egg method takes around 7 months to produce. This gives plenty of time for the viruses to change outside in the world. Because of this, the vaccines being produced in labs may not be able to properly deal with the newly mutated strains. Another problem is the fact that the vaccine virus can mutate while in the incubation process. This means that a vaccine may not work as intended once it's purified and sent out to distributors. Ferrets are also a problem. Ferrets are very commonly used to find a strain to create the vaccine out of. They do this by injecting the strain into a ferret, collecting the ferret's antibodies, and testing them out on a human strain of influenza. The problem with this is the fact that ferrets are not humans, and just because the ferret antibodies worked does not guarantee that the vaccine will work with humans. There is also a lot of unpredictability when creating flu vaccines. Researchers have to predict which strains will affect humans during a given flu season. If they predict wrong, the vaccine will be less effective. There is also no guarantee that the produced vaccine will work with every person. This chart provided by the CDC gives the percentages for flu vaccine effectiveness over the years of 2004 through this year. The CDC gives an average of about 40 to 60% effectiveness of flu vaccines. The efficacy, or the effectiveness of the vaccine under controlled conditions, is typically very high for the flu vaccines, but the real-life effectiveness is usually much lower. The vaccines typically work better with influenza A, H1N1, and B viruses, and worse against influenza A, H3N2 viruses. The effectiveness varies greatly depending on the type of virus. The effectiveness is also dependent on the health of the person being vaccinated, with healthier people typically seeing better results. Some studies showed the live form of the vaccine may actually be more effective than the traditional inactive method.